Okay, we're going to go ahead and get started. Our next speaker, I'm just going to say her name because she told me not to do her introduction. This is Dr. Carol Thiele from the NCI, and I asked her to talk about the role of NMYC in diagnosing, treating, and the outcomes of neuroblastoma. Dr. Carol Thiele.
Is my mic on? Okay, there we go. And the reason I was going to tell you a little bit about my journey to neuroblastoma, because anyway, well, that's just the way I thought I would start in. So I'm going to talk about the role of MYCN in neuroblastoma tumor biology from kind of a historical perspective. And I also want you to get a feel for where science was when we first started this journey and how far we've come. So first of all, I have to give you a little bit of background about, you've heard the word NIH and NCI, but I don't think a lot of people really understand what this is. And what this is your tax dollars being paid to support biomedical research across the country. The majority of true biomedical research is being supported by the government. And so the National Institutes of Health is a whole series of smaller institutes that are dedicated to various aspects of health, and I'm in the National Cancer Institute. Now, the National Cancer Institute is the largest of all the institutes, and I see Pat's going to be checking me because it comprised two parts. There's an extramural program and an intramural research program. And the extramural program funds all the grants for that the university professors and researchers get through R01 type mechanisms, K awards for young investigators, or program projects like the big neuroblastoma P01 grant. It also funds programs like the Children's Oncology Group and their research, as well as cancer centers. A small piece of this pie is then dedicated to what's called the intramural research program. And we have what's called the Center for Cancer Research, and that's where I am. The other part of it, the little smaller part, is the Division of Cancer Epidemiology and Genetics. So this is how NCI, and it's much more complicated in the extramural. I've just given you a slice of things that may resonate with you. So the Center for Cancer Research is located in Bethesda, Maryland, and this is a photo of what is the Center for Clinical Research, which is the largest hospital dedicated to research. So patients come to this research center for clinical trials, only clinical trials, and predominantly they are treating and looking to treat and explore diseases for which there are no known causes or no known cures. The Center for Cancer Research has 23 basic science laboratories and 17 clinical research branches. And I am in one of the clinical branches called the Pediatric Oncology Branch. And this is a group of clinical and basic scientists, as you can see here. Oh, it's not working, the little dot. Oh, there it is, okay. So we have a number of investigators that span from basic, as you can see my name down there with the yellow star, all the way to clinical. And the two stars are the two groups that are really focused on neuroblastoma. So my group, it says cell and molecular biology, but we're really focused on neuroblastoma. And then Rosa Nguyen's group, who's come as a young tenure track investigator to look at immunotherapy in neuroblastoma and CAR T therapies. So when did I first come to learn about neuroblastoma? It was when I was a graduate student at UCLA. And there, in a newly developed clinical immunology department, Bob Seeger was making monoclonal antibodies to immunizing mice with neuroblastoma cells, trying to find antibodies that would specifically recognize the tumor cells. And this was the strategy that Pat used and many other researchers used that led to the development of the anti-GD2 antibody and Pat's HSAN antibody as well. So this was a strategy that the immunotherapies were trying to use. But what I saw, even though I wasn't really studying neuroblastoma at the time here, I was studying B-cell development. Sometimes it's working. Was Neil Seidel, a postdoc who was working with Neil Seidel, who had found that he could take neuroblastoma tumor cells, treat them with a derivative of vitamin A, and get them to stop growing and turn into what looked like normal neural cells. And this to me was fascinating. So you have a tumor cell turning into a normal cell. And this stayed in the back of my mind all the while. I went later on I came to the NIH to study molecular biology or gene cloning. So I was fascinated by the fact that when I learned more about neuroblastoma, there were a number of clinical clues that indicated that there were anomalies in development that contributed to neuroblastoma. And here you see the histology of 3 different types of neuroblastoma: the small round blue cell tumors of neuroblastoma that have a less differentiated phenotype, and the spectrums that you see where you have a mixed phenotype, as well as the really well differentiated. We now know that this is favorable histology, and that these are linked to low-risk disease, intermediate, and these are high-risk disease. So it's the undifferentiated ones that tend to be linked more to high-risk disease. And it was Pat who first taught me how to treat neuroblastomas with retinoic acid, and my charge was to find out what were the genes controlling the growth and differentiation of these cells using this model system. And it really was a terrific system because it was very gratifying. At least for some neuroblastoma cell lines, it always worked. There's lots of information to understand too later on why some neuroblastomas don't respond to retinoic acid. An equally interesting question, but we were focused on what were the genes that were controlled. So trying to understand genes that were controlling this process. And what I like people to think about is, everybody understands an egg and a sperm. These are highly differentiated cells. Every cell in your body has the same number of genes. But you take this egg and this sperm, and when they come together, these highly differentiated cells now come together to form this new unit where all the genes now are open to be expressed. And it's the selective differential expression of these genes that give rise to all the different cell types in your body. I mean, it's really mind-blowing that this kind of thing happens, and that it happens so well most of the time. So one of the things that was known at this time was that there were chromosomal anomalies in neuroblastoma cells. These, they look like kind of worms, but are actually banded chromosomes, and people could identify each of the different chromosomes, but they noticed that some had these homogeneous staining regions, and in other types they had these small double minute chromosomes. And these were the chromosomal manifestations of gene amplification. And so the really important point was to try to understand what gene is being amplified in neuroblastoma. So Manfred Schwab found that it was a gene that was related to a protein MYC that that was isolated from chicken virus that caused tumors in chickens. And this gene that was related to this, what we termed viral oncogene, was then named neuroblastoma MYC. Because you can see here, when we look at this 2 kb fragment is only expressed in neuroblastoma cells and not in other cells. Because neuroblastoma patients are treated in a cooperative manner, there were a large number of tumor specimens that people could then look to see, is this happening in all the tumors or only some of the tumors? I'm having problems changing. Oh, there we go. So Gerard-Brodeur looked at the DNA from tumors of patients with neuroblastoma and then found that there was a percentage of patients that had this band that was indicative of multiple copies of this NMYC gene. And they later found that the association of multiple copies of this NMYC gene was associated with rapid progression of the disease. And this is really where MYCN was first appreciated to be a biomarker of risk. So while we were studying what were the genes that were involved in controlling growth and differentiation, it was really a natural experiment to look to see what was happening with retinoic when we treated cells with retinoic acid to the NMYC gene. And what we found was that if you decreased expression of NMYC, this decrease preceded morphological differentiation. It suggested that the differentiation link or defect in the neuroblastoma cells with this amplification was linked to the expression of this gene. So that was sort of the background knowledge that we had when Pat convinced the COG to begin to study retinoic acid in a clinical trial. And he used 13-cis retinoic acid because that was clinically approved for skin. And this was the results. So you can see that we identified this in 1985. Retinoic acid was the first agent that you could think would target NMYC. And in point of fact, the trial was initiated in the 1990s, and then the results came out much later. And what was interesting, I found, was the fact that if you look at these graphs, the patients got the chemotherapy with or without retinoic acid, or a bone marrow transplant with and without retinoic acid, as well as anti-GD2 and cytokines. And if you think that the retinoic acid was only working on the tumor cells and inhibiting the growth of the tumor cells, you might think that it should work in this context as well. This really said that, in point of fact, the immunotherapy was very important. The bone marrow transplant and the immunotherapy was very important. It was really the first indication that there was an interplay between the immune system and what was going on with this kind of therapy and the importance of this therapy. So I continued on with these kinds of thoughts in mind in my studies. I try to understand what was the role in this. So we had evolved technologies at this time where we could specifically go in and silence genes in cells. And we do this with what I called silencing DNA or RNAs. And you can see here, when we silence MYCN and look at the growth of the cells, there's a decreased growth. And in fact, if you silence NMYC, now you can see the cells can elaborate a differentiation profile. So it also increases the loss of NMYC, increases the ability of the cells to differentiate, or removes a block that they have to differentiate. So this is the model that we were working on, that NMYC stimulates self-renewal in the cells and blocks differentiation. and that cis-retinoic acid blocks this effect of MYCN. Now, I want to just give you, at the same time, a mindset of how we were thinking about how normal cells grow. So there are growth-stimulating genes that help cells proliferate, and there are growth-suppressing genes. And this is a simplistic model, but then how does a normal cell become a tumor cell? And at the time, it began to be the interplay of these two types of activities. You needed something as a growth-promoting gene, and you needed something to a tumor suppressor gene, the loss of a tumor suppressor gene. It's a little bit like driving a car. You need an accelerator to get it going and a brake to stop it. You can drive with both of them on, but you go a lot slower. And it's a little bit what happens with a tumor cell, that once you relieve the brake, you can really go. The other part I want to talk to you about is how the cells differentiate. So cells tend to have a progenitor cell. Remember the fact that they're undifferentiated. And neuroblastoma comes from a neural crest cell. And the neural crest cell can give rise to many different kinds of cells. It gives rise to the pigmented cells in your body if it travels during development in one way. It gives rise to some of the craniofacial bony structures. Some parts of your vascular system are neural crest derived, as well as what you saw earlier, that it gives rise to the chromaffin cells of the adrenal gland and all the sympathetic neurons. So it's a tissue that's highly plastic. And it's really responsive to environmental signals and cues, and that these enable the cell to change its state or the pattern of gene expression. And in a tumor cell, what we think is happening, and especially with MYCN, we think that MYCN actually is, you have a persistent self-renewal, and you have an inability to differentiate. And so the cells stay in this undifferentiated state because they don't have enough of the signals to get them to transit into a differentiated state. And during development, you can see that actually, as they differentiate into different lineages, you would also create barriers so that you can't, your nerve cell can't become your skin cell or your eye cell. So there are barriers once the cell has committed to a lineage. But we also think that neuroblastoma cells have a high degree of plasticity, which means that the barriers to transit these cell states has been lowered, so they can switch back and forth at a much higher propensity. And I come back to this because I was always intrigued about the association between MYCN amplification and the loss of 1p36, loss of heterozygosity, or the loss of one allele on chromosome 1p, around 1p36, that you see this association. And this association was associated with unfavorable or undifferentiated histology. And so why is there always an association between these two things? Sorry. So during development, one of the major systems that can help cells determine their lineages and keep their lineages intact is something called polycomb repressor complex. And its key enzymatic enzyme is EZH2, which really, once this is highly expressed and particular marks are made around the genes, suppresses the ability of these genes to be expressed because the DNA doesn't become accessible. And so you can see here that in neuroblastomas with unfavorable histology, they have really high levels of this. So it's preventing the implementation of a differentiation program. So we began to study this, And we used a model system where we could look to see how NMYC and this EZH2 were related. So we can transfect the neuroblastoma cells with a vector that can suppress MYC expression with a drug, tetracycline. When we take it away, now you can see that MYCN gets expressed. Wow, this really changes. And as you can see with MYCN, as MYCN goes up, eventually with time, EZH2 goes up, and this histone mark that it methylates, it goes up as well. And then a gene that's on chromosome 1p36, CASI1, that we've studied, is suppressed. So we showed in this study that there's EZH2 dysregulation in neuroblastoma, and it inhibits a number of different genes that have tumor suppressor activity or enable a cell to make a differentiation program. And then later in another study, we showed that if you take a small molecule inhibitor and treat the cells, you can inhibit the growth of the neuroblastomas. So it gives us a model in which we think that MYCN is stimulating EZH2 expression, and this can block a differentiation gene. And if you can inhibit this, then you can also affect the differentiation. EZH2 also does another thing. It has recently been shown to impact the levels of GD2 expression in some low-expressing neuroblastoma cells. And if you use an inhibitor of EZH2, you can get re-expression. It also seems to be involved in MHC class 1 expression. So neuroblastomas have low levels of class 1 expression, as if you recall earlier from the talk. This is one of the molecules that your immune cells need to recognize on the different cells in order to distinguish something that's self from something that's foreign. And in this study, it was shown that if you treat the neuroblastoma cells, you can increase the expression of MHC2 class 1 antigens. And in this way, then, you could actually make the cells be better recognized by immune cells. So in the last part, I just want to talk a little bit about how MYCN works in different cell types. So these are some studies that we've recently done in which we've taken patient-derived cells and put them in a culture media, and you can see they grow as spheres. When we shift them to another kind of media, you can see now we have two different kinds of morphological cells. We have a very flat cell and a very round cell. This had been known since the 1980s that, in fact, you had substrate adherent cells and you had cells that look more neuronal. More recently, they've been called based on all their gene expression. So the genes that are more neuronal have an adrenergic transcriptome, while the cells that are more substrate adherent have a more mesenchymal. What is interesting is the fact that this is from a MYCN amplified tumor. The cells all have MYCN amplification, but the mesenchymal cells here have silenced the expression of MYCN. If we look at a patient's tumors, there's a very small percentage of cells in patients that actually have markers associated with mesenchymal cells, okay, or these phenotypic cells. And we've modeled these, and when we put them individually into tumors, the mesenchymal cells take a very long time to make a tumor, and when we look at the tumors at the end, they look more adrenergic. So this has enabled people to talk about the fact that the cells can phenotypically switch. And this relates to the plasticity that I was talking about. We also know that if you treat the cells with chemotherapy, a lot of the cells that remain are have more mesenchymal phenotype. And so, are these the cells that maybe give rise to the resistance that you see? People have talked about identifying targets in mesenchymal cells, and we've done a little bit of work in it. Here is our two cell types, mesenchymal and adrenergic, looking at a recent target, GPC2, there's a clinical trial up at CHOP, a CAR T trial targeting GPC2, and you can see that it's mainly the adrenergic cells that are expressing this. In the same way, mainly the adrenergic cells are expressing GD2, while the mesenchymal cells in this model have very low levels. So we performed some mass spec analysis and looked at the surface proteins of the mesenchymal cells and the adrenergic cells to look for targets that might not be, that were differentially expressed. The third time's the charm. So here you can see in this map where the darker blue means it's more highly expressed, that a protein like PTK7 is highly expressed in the mesenchymal cells, and it's also expressed in the adrenergic cells. And so when people asked about earlier in low GD2 cells, it's possible they can express PTK7. And Kelly Goldsmith at Aflac Children's Hospital has she has a T-cell receptor targeted therapy using PTK7. And we've modeled PTK7 as a target using CAR T cells, and it's very effective, and we can have evidence that the combination of something like a GPC2 and a GD2 is better than either of them alone. So the last thing I just want to share with you is that we've been evaluating drugs to try to understand this phenotypic switching. And using this model, you can see that when we take the cells and move them into fetal calves, you can see the appearance of these red mesenchymal-like cells, and then the yellow, more adrenergic balls of cells. If we do single-cell analysis of this population, we can see that when they're grown in one media, there's an enrichment, a big enrichment when we shift to fetal calf serum in the mesenchymal population, consistent with the appearance of these cells. And so we've been screening, looking for drugs that will block this conversion. And one of the drugs that we've identified is an inhibitor of a protein that helps remodel nucleosomes, called a SWI/SNF ATPase inhibitor. And you can see that when we shift the cells into media that contains this inhibitor, we don't see the appearance of these mesenchymal cells. And when we look by single cell, there's a real decrease in cells expressing this mesenchymal phenotype. And remember, I told you the adrenergic cells were more sensitive to chemotherapy. And so, in point of fact, if you then combine using this inhibitor with etoposide, you can see in this confluence growth at the bottom, this is a very effective combination. So this really is it's just going to be published. This is really preclinical, but it's to share with you the idea that the researchers are really at this idea that we're trying to understand potentially how resistant disease develops, the strategies that we're using. I wanted to get back to this final graph and just say, I think even though we didn't know why this worked, and we had models to think about, we could have proposed, I think there's lots of things that possibly are happening. The cis-retinoic acid decreases the MYCN, it decreases the EZH2. This may enable re-expression of class 1. In point of fact, you could have now the engagement of T cells. And I think this is the result from the most recent where tandem transplant and cis-retinoic acid with anti-GD2 and cytokines is really shown to be an effective therapy for MYCN-amplified tumors. But we know that it's not good enough. We know that this therapeutic approach is still too toxic for many patients, and it's inadequate, so some are non-responsive. And more importantly, for those that we actually can help, there's long-term morbidities, as well as the potential for relapse. And so I think that all the investigators are working at different aspects of this, from both the basic as well as the clinical. And I think the investment that the American people made in biomedical research has really accelerated this. I know many of you are too young to know when Nixon, he said, when we landed on the moon, he said, if we can put a man on the moon, we can cure cancer in 50 years. The problem was this analogy was false, because it would have been if we had put a man on a moon where we didn't even know where the moon was going to be. Because at that time, we didn't know how a normal cell controlled its growth and differentiation, let alone how a cancer gene did that. And so I think that we're in an exponential phase of our understanding of these systems, and I think the ability to translate has been contracted, and I think this has really been at the urging of all the parent groups who've been and advocates who've been involved in this, because they've helped change the philosophy at different government institutions in terms of how trials should be. I know we at the POB are very lucky because our branch is really aligned with the adult oncologist. So when they have a therapy that we think is going to be useful for children, we can piggyback children into the trial almost immediately. And so this has really helped to be a way to accelerate the development of getting therapies into our pediatric population sooner. So with that, I just want to share the work done by my team. Really, Jui Liu leads my lab as I've moved more into administrative things. And I want to tell you that when I first came to neuroblastoma meeting, there probably were this many people at the meeting, scientists and clinicians. And next year, we will have ANR in Washington, D.C., and we expect to have over 800 international clinicians and scientists coming to study this disease. Thank you.
Thank you. Does anyone have any questions? We're all NMIC experts. 30 years, I still don't get it. Anyway, no one has questions? Okay. Thank you, Dr. Thiele.